I just hold that, or I put it. Just can put it on the neck like that. Okay. Hello, thank you, Munzer, for that very nice introduction. So, yeah, it's a pleasure being here, and thank you, Devarat, for inviting me. It's really nice because when I was at MIT, I had the the privilege to work with amazing faculty who have done amazing things in electrical engineering, from communication systems and control, and many of them are here as speakers. Dr. Warnell was we say doctor at Hopkins now. Professor Warnell was actually my academic advisor. Munzer was my PhD advisor. Sanjay Mitter, who's coming later today, was on my thesis committee. So it's just really, really nice to see everybody here. So because I'm giving a short course, I think I'm talking in the morning, talking a little bit in the afternoon. And just to give you an overview, I'm assuming that most people have not been exposed to neuroscience. So I'm going to give you sort of the minimal amount of information that you need to push through everything that I'm going to talk about today. And what I want to say is if any of this is interesting to you, especially the PhD students out there, there's no question with people from our backgrounds, with this training of systems control, dynamical systems modeling, we can make very interesting contributions in the field of neuroscience. And in particular, neuroscience is all about trying to understand brain function. And the types of techniques we have are just not there in the broad neuroscience community. And so we can really, really solve important problems. Okay, so I'm going to walk you through. The top half is in the morning. I'll talk about just briefly global initiatives in the United States and Europe in terms of brain science. I'm going to give a crash course. I call it Neuroscience in 15 minutes. Hopefully you'll appreciate the complexity of neuroscience. For those who don't know, if you go to the main annual meeting of the Society for Neuroscience, it's an international meeting. There are 35,000 people who come every year. So in the United States, that can only be in one of 3 cities because it's so big. And that should just give you an idea of how complicated this field is. Because when somebody's a neuroscientist, they tend to be an expert in one particular area out of thousands. And then once I do that, I'll talk about data, of course. I want to make sure you understand the type of neural recordings we're talking about. And I'm going to move right into modeling, how we can build models to understand brain function. And my first application that I'm going to talk about this morning is gambling, decision making. So I'll talk about a study we did where we look at humans making decisions and recording from their brain. And then in the afternoon, I'm going to move on to movements, motor control. This is more of a control theoretical analysis of sensory motor control systems. We're going to talk about trade-offs between brain and muscle when you try to track fast movements. And then finally, I hope time permits, I'll talk about sort of EEG networks in epilepsy and some of the work we're doing to translate into the clinic. Okay, and stop me at any time if you have any questions on anything I'm saying today. So many, many years ago when Obama was president, he launched the Brain Initiative in the United States. And basically $100 million, which is not a whole lot at the end of the day when you split that between 35,000 people, but it was an effort to bring in more and more resources for people to study the brain. And so he says it really nicely here in this quote. He says, as humans, we can identify galaxies light years away. But when it comes to the brain, which is just 3 pounds of matter between our ears, we still are very much in the infancy in terms of understanding brain function. So global initiative. So with this brain initiative, we launched a couple of projects. In the United States, it was the Human Connectome project. Essentially what this is a big challenge to create a full wiring diagram of the brain. So just wires. Not physiology, not function, but just structure. Who connects to who? And how strong are these connections? So that's the Human Connectome project. We have Blue Brain, which is from Europe, which is essentially trying to build a massive mechanistic model of the brain, neuron by neuron. Okay, so it's going to have billions of neurons, and each neuron is modeled with some nonlinear ordinary differential equation that characterizes the biophysics of how neurons fire action potentials, and just try to build a simulation environment of the entire brain. Okay, so here's the crash course. Yes? Where they stand? Well, the Human Connectome, for certain, like C. elegans and certain smaller creatures, we actually have full wiring diagrams. But for the human brain, we're really trying to get at a global database, almost like an atlas. I wouldn't say we're there yet, but we're getting very close. In terms of blue brain, I'm not exactly sure how much they have, but they definitely have parts of the brain entirely coded up, like visual cortex. I'll talk a little bit about it today. So certain regions of the brain and how they process stimuli and generate electrical activity, that's where they're at. But this is all online if you wanted to see where things are at.
So in the first one, do you want to just build a model of the thing, or do you want actually to know how intense the connections are, or what
Right. So the latter. So they're interested in this wiring diagram so that people can then study what happens, say, when you go from development, from childhood to adulthood, so you can understand how these wiring strengths change, the diagram changes over time. They're also interested in diseases. So basically, it's just an imaging technique to get this wiring diagram. It's called diffusion tensor imaging. You take different imaging modalities, you combine them using an atlas, and then you draw the wires. Okay, that's essentially what it's doing. And they're interested, for example, in disease. If you have Alzheimer's, they want to understand, okay, at the end of the day, is the structural wiring diagram affected by disease? And if so, how? Which regions are affected in terms of the strength of the connection? Is it global, across the entire brain, or within a particular network? Okay, and if you know that, then you can start targeting drugs specifically to different networks, because the networks, even though they're structurally different, they have different chemical properties, neurotransmitters around that govern the activity in those regions. So you can actually target, therapies if you find out what's going on. Yeah? Okay, so essentially you might remember from maybe high school, you have essentially 4 lobes in the brain. You have a frontal lobe, so this is just basics anatomy. Cerebrum, which is cerebral cortex, which is contrast to the cerebellum, which is sometimes called the mini cortex. That's not actually a lobe. I'll talk about what general function in a second. You have frontal lobe, parietal lobe, temporal lobe, and in the back, occipital lobe. Okay? And then you have something else, cerebellum here, and then you have something else called the midbrain, and then the brainstem, which then connects down to your spinal cord. Okay? And so the traditional way we used to study function, or we still study function of the brain, is to find out what goes wrong when it breaks. Okay? That's kind of how complicated it is. So I'll take you through some basics, what we learn in Introduction to Neuroscience. Okay? So if you're talking about the frontal lobe, essentially this is sort of higher cognitive processing. So decision making, planning, motivation, inhibition, and so forth, attention, all is definitely regulated minimally by the frontal lobe. It's not exclusive to the frontal lobe. Just to give you an idea of inhibition, for example, when a child is born, the frontal lobe is not fully developed. And in fact, evidence today says your frontal lobe is not fully developed until you're in your early 20s. So I don't know if you have children, toddlers, right? And sometimes toddlers say inappropriate things, right? They might say something that the parents might have criticized a neighbor, and then the child will go off and tell the neighbor, right? Why do they do that? Because actually they don't have their frontal lobe fully formed. And so they don't have the inhibition, the filters of, okay, is this an appropriate thing to say in this social context or not? That's definitely frontal lobal function. So when it goes wrong, you see problems. In personality at the end of the day is what's effective, but you have inappropriate responses, you might lack motivation, and a complete personality change. And this is a very famous person to hit the nail home in terms of frontal lobal damage. So this is Phineas Gage. This is the very first person you learn about when you're a neuroscience student as an undergraduate. So Phineas Gage was a construction worker in the mid-1800s of the United States. So in the United States in the mid-1800s, it was the railroad revolution, okay? So the railroad industry emerged, and people were laying railroad tracks to go all the way from the East Coast to the West Coast of America. And so when you lay down, so Phineas Gage was laying down railroad tracks. And when you lay things down, sometimes you have to blast rock, okay, with dynamite. Okay, so they have these lead rods and they're blasting rock. It's quite dangerous. And what happened to Phineas Gage is when he was blasting rock or working in the railroads, there was an explosion that happened, and his own iron rod flew under his left eye, pierced his brain, and flew out about 100 feet away from himself. In fact, here's sort of the hole that you see from the skull. So the trajectory was it went under the left eye, through the left frontal lobe, and out. and it actually shot out hundreds of feet, which is kind of remarkable. But what was really amazing is that he was completely conscious. Okay, he wasn't dead. Not only was he alive, but he was able to get himself onto a horse wagon and get treatment. The second miracle was he didn't die of an infection. So he actually survived this entire incident. So people thought, wow, I mean, and when he's, recovered, he seemed in the beginning sort of normal. All cognitive function was intact in the sense of computing things. He was able to compute, he could play bridge, whatever, okay? But what did change is sort of his emotional processing and decision making. So no longer was he able to make decisions that were good for his own self or survival. He would say inappropriate things in public, and actually he couldn't hold a job anymore. So initially when they survived, they thought, well, maybe we don't really need that part of the frontal lobe because he looks normal. But then over time, they saw the behavioral changes and then started associating sort of personality, emotion, decision-making to frontal lobe. Okay, so parietal lobe. So integrate sensory information, that's loaded. Okay, what do I mean by integrating sensory information? Where we have all our 5 senses, touch, smell, vision, and so forth. And somehow as you're moving around the environment, you're getting all kinds of stimuli from all these different modalities. And the parietal lobe, yeah.
So how, the person
yeah.
It's not like how do you single out that because of the damage of
Mm-hmm.
it could be because of the
Well, so this is what they changed. So this is before to after. So before, he was a very reliable worker, made good decisions, personable, had lots of friends. And then after this accident, he couldn't keep a job. He couldn't make good decisions. He wasn't reliable. Social etiquette changed. So this is what I mean by how the traditional way of studying the brain is you just see what happens when there's a lesion or a damage in terms of a change in behavior. And then they associate that back to the function of the brain. But it's not true. I mean, one of the things that I hope as I go through some of the studies today is to have you appreciate that the frontal lobe, it has lots of dense connections coming from midbrain, primitive areas that deal with emotion, visual cortex, and so forth. All of these regions are connecting to frontal lobe. So you cannot ever say that this particular part of your brain is governing rationality. It's just false. Okay, does that get at what you're saying? I mean, actually, my father-in-law in 1992 got into a very severe car accident in Zimbabwe in Africa. He was just sitting in his car, and a fire truck came at a very high speed, and he went head forward through the windshield. Okay, and he thought the doctors thought he was going to die within a day or two. He miraculously survived, but if you look at an MRI scan, he has no frontal lobe. He actually lives in the United States. He can't drive and so forth. He has no frontal lobe. Now, I only met him after that, so I don't know what he was like before, but if you ask my mother-in-law, she said that all of a sudden, one day she woke up and she's married to a completely different person. His personality changed. He jokes a lot. He used to say inappropriate things in the beginning, but I guess over time, as he was sort of recovering, the brain rewires. It's really, really robust in the sense that even in adulthood, your brain will rewire if there's damage. But personality, no motivation. I think, their neighbor's house was burning down, and the whole neighborhood got out of their house concerned. He just went right back to sleep. He didn't care, right? These are things that he wasn't like that before the accident. So there's very profound changes when you have frontal lobal damage. So parietal lobe has to do with planning. So we'll talk about movements later on today, but you're going to take all this information and you're going to think about, okay, how do I want to navigate around this world? The parietal lobe has a lot to do with the planning of that navigation. It also, depending on the left versus the right side of the parietal lobe, you talk about numbers, relations, manipulations of objects, tend to be encoded in this region. What happens when you have damage? Well, it depends on whether you're on the right side or the left side. You don't have to read all of this, but you might have loss of imagery, visual spatial relationship, hemispheric neglection of the left side of the space. So I think there was an old Seinfeld episode, I don't know if any of you, I'm dating myself, of Seinfeld, where Kramer walks in and he had his pants on one side of his leg and not the other side. That's this neglect. It comes from right parietal lobe damage. You don't see the left side, okay, or you don't perceive it. Damage to the left has problems with mathematics, reading, writing, and so forth. The left side of the brain is definitely governing language, not entirely, but dominantly related to language. Occipital lobe, back of the brain, contains your visual cortex. So this is where all visual stimuli are perceived, in the back of the brain. Okay, it contains low-level descriptions of orientations of objects, color, size, and so forth, and you have very, very small receptive fields to allow you to really perceive small changes in contracts and edges and so forth in orientation. So of course, if you have damage to your occipital lobe, you're going to have all kinds of visual disabilities, maybe even blindness. So, you know, for those, I mean, I guess for children, when you talk about this, you can actually have completely intact eyes and retinas and optic tracts, and they can be fully functional but still be blind. Why? Because when the visual stimulus comes into your eye and it goes down through optic tract, it ends, well, it doesn't end, but it hits the back of the brain, which is visual cortex. At that moment, that's when you perceive the image, okay? And if you don't have a visual cortex or you have damage, even though all this image, the light is coming into your eye and into your brain, you're never going to perceive it because it's of the visual cortex damage. Okay? Let's see. Temporal lobe. Okay, temporal lobe is sort of in the green as you see. So this definitely has to do with memory. Okay, a big structure called the not big, but a dominant structure within temporal lobe is called the hippocampus, and that's where lots of evidence shows that memories are retrieved, they are stored, and so forth. So you have a lot of memory storage from all types of modality, whether it's visual memories, auditory, olfaction, smell. Things tend to be stored and